Hello everyone, and in this class I'm going to discuss about the protein structure and its function. So first, let us discuss what is protein. So protein are most abundant molecules of living system. So we know in our human body or in animal body, its majority of the component is protein. So in 1939, Dutch chemist Gerdius Johannes Mulder was first who discovered the protein. So protein comes from the Greek word proteos, which means the first place. So proteins are the important class of biological molecule which are the polymers of amino acids. So protein are most essential building block of human body. Okay, so we can see the pictorial representation of protein. What is protein? So you can see that amino acid is forming peptide by amide bond formation, and then peptide is giving the protein. So let us first know what is amino acid. So this is the building block of protein. So these are organic compounds containing two functional groups on carboxylic acid. Another is amino group. The carboxylic group is acidic in nature, while the NH2 is basic in nature. So each amino acid has four different groups which attach to alpha carbon containing side chain R, a carboxylic group, a NH2 group, and a hydrogen atom. So peptide bond. This is also important to know what is peptide bond. So the covalent NaCONH bond is formed between the alpha carboxylic acid of one amino acid with side chain, okay, R1, and alpha amino group of the another amino acid with side chain R2. You can see in the picture that the bond is forming between the carboxylic group of amino acid and NH2 group of another amino acid. Okay, and then it is forming and after elimination of water, a molecule of water, it is forming a COONH bond. This is called amide bond or peptide bond. Okay, so if three amino acid units are there, then we call it a tripeptide. If it is more than 10 amino acids are forming these peptides by this amide bond formation, we call it oligopeptides, and beyond 10 we call it polypeptides. Okay, so when polypeptides made of over 100 amino acid, it is called macropeptides. So structural organization of protein. So this is very important to know what are the structure of protein. So primary structure, secondary structure, tertiary structure, and quaternary structure. So primary structure means the sequence of amino acid. Okay, sequence of amino acid in a peptide chain. Okay, so primary structure of protein refers to sequence of amino acid in a peptide chain. It was discovered by Fischer in 1906. Amino acids are linked together by peptide bond. Each amino acid is in a polypeptide is called residue or moiety. The primary structure of protein starts with an amino acid group NH2 group of N-terminal end and ends with a carboxylic group of C-terminal end. So if you can see in the picture, so this is the primary structure representation of protein. So it's a peptide chain, and we can see those are the peptide bonds, and those pink are the amino acid. So this sequence which amino acid will attach with which another amino acid and what kind of structure it will form. So primary structure deals with the sequence of amino acid. So you can see here, okay, this is the peptide bond. So you can see that this is the meta alkophalin. It's a painkiller available within our body or human body, natural painkiller. This is the overall structure. So we can see that a tyrosine is attaching to glycine, and then again glycine, phenylalanine, and methionine. So this is how this protein moiety, protein or drug molecule is peptide is formed. Okay, so this arrangement, tyrosine, glycine, phenyl, methionine, this is the primary structure of the protein. The secondary structure of protein. So arrangements of adjacent amino acid forms polypeptide chain folds. It consists of maybe alpha helix, beta pleated sheets, and triple helix. So what is alpha helix or local folding or alpha conformation? So, based on the hydrogen bonding, Linus Pauling and Robert Corey in 1951 identified alpha helix and beta pleated structure. It is the spiral staircase structure. You can see here it's a spiral staircase structure, tightly packed and coiled polypeptide chain. It is stabilized by hydrogen bonding between the different nucleobases present in the protein or polypeptide chain. Okay, so amino acid per unit per turn is 3.66 in average, and the distance between one strand to another turn is 0.54 nanometer. So glycine and protein are alpha helix breaker and produces bend in the helixes. So for example, keratin and myoglobin. The side chain R group of the amino acid in the alpha helix extended outward from the helical backbone. This outward orientation minimizes steric hindrance between the R group. So occurrence. So alpha helixes are a common structural element in proteins. They can be found in various regions of the protein, including transmembrane domain, DNA binding region, and structural framework. So helix destabilizing amino acids, as I told, that proline and glycine is the helix breaker. Okay, glycine and proline. So proline as a helix breaker, since nitrogen of proline residue is a peptide linkage, has no substituted hydrogen, because we know that this is secondary amine group present in proline. Okay, for the formation of hydrogen bond with the other residue, proline fits only the first turn of the alpha helix. Elsewhere, it produces bend and turn. And glycine, we know glycine is the smallest amino acid because side chain is hydrogen. So all bend in alpha helix are not caused by proline residue, but bend often occur also at the glycine residue, as the side chain of the glycine is the very small. So it will make a bend in the protein structure. What is beta pleated seed? So this structure are formed when two or more polypeptide chains line up side by side to form a zigzag structure. These are stabilized by hydrogen bonding. Okay, it is fully extended. Adjacent amino acids are separated by 3.5 angstrom. These are further classified as parallel beta pleated sheets and anti-parallel beta pleated sheets. So this is, we can see parallel beta pleated sheet. This is anti-parallel. When both orientation of the peptide chain in the same direction, we call it parallel, and when it is opposite, we call it anti-parallel. So anti-parallel beta sheets, you can see the orientation of the two polypeptide chains in the opposite direction, and this kind of structure is found in silk fibrin. And parallel beta pleated sheets, the orientation of the two polypeptide chains in the same direction, and this kind of structure found in beta keratin. So this is the overall representation of the protein secondary structure. So this is the alpha helix, you can see helical structure, and this is the beta pleated sheet. So you can see two polypeptide chains are lining up next to each other and making the hydrogen bonding and forming this kind of beta pleated sheet. Triple helical structure. This is also a very important structure. Okay, so it is in principle structural element of a human body and make up to 25 to 33% of the overall body. So it is found in the connective tissues such as tendon, cartilage, and organic matrix of bones and cornea of the eye. So these three polypeptide chains are twisted around each other like forming a rope from the thread. Okay, so for example, collagen. Collagen is a main structural protein in various connective tissues in animal, making it most abundant protein in mammals. So this is the pictorial representation how the collagen is formed. So you can see three polypeptide chains. They are uh, mm, uh, they are making a spiral structure and making the collagen. So what is tertiary structure of the protein? Structure is a three-dimensional conformation of the polypeptide. The tertiary structure of proteins reveals about the biological function of protein. The function of a protein is depend upon its tertiary structure. If it is disrupted, it loses its functional properties. What are the forces responsible for stabilizing the tertiary structure? So the final shape of protein are determined by different bonding interaction between the side chains of amino acids. So what are those interaction? It could be hydrogen bonding, it could be ionic bonding, it could be disulfide bridges, it could be hydrophobic interaction. So in the picture, you can see different kind of interaction which are responsible for stabilizing the tertiary structure of the protein. So you can see there is a possibility of formation of hydrogen bonding. So there is a hydrophobic interaction, okay, between the two group side chain, and there is a sulfur-sulfur bond, that is cysteine. In case of cysteine, amino acid is possible, and there is an ionic interaction. It could be between the carboxylate and the NH3 plus ion of two different peptide in this peptide group, two different groups. So these are the different forces which are responsible for stabilizing the tertiary structure of the protein. So now let us talk about the quaternary structure of protein. So many proteins are made up of multiple polypeptide chains and are referred to as a protein subunit. The quaternary structure refers to the interaction of the protein subunit with each other and arrange themselves to form large aggregate protein complex. It involves the cluster of several individual polypeptides or protein into final specific shape. So a variety if of bonding interaction holds the various chains into particular geometry. So you can see this is on polypeptide chain. This is another one. So both are combining and making a very complex quaternary structure by several kind of interaction. Those are the common interaction: hydrogen bonding, ionic interaction, hydrophobic interaction. All the interaction are holding this structure and making the quaternary structure. Depending upon the number of polypeptide chain, it may be dimeric containing two polypeptides, as for example insulin, tetrameric containing four polypeptides, as for example hemoglobin, and then we can see that two-dimensional alpha chain, it consists of 141 amino acid, two dihydroxy beta chain, 146 amino acid. So these are the examples of this quaternary structure of the protein. So now let us discuss about type of protein. So types of proteins. So proteins could be two types. One is fibrous, another is globular protein. So fibrous proteins is a long and narrow. It is generally soluble in water. It contains a repetitive sequence of amino acids. It plays a great role in strength and support. It is less sensitive to change in pH and heat. For example, keratin, which is found in our hair or nails, and elastin, myosin. Okay, so you can see the collagen. This is an example of fibrous protein. And globular protein. So it is rounded and spherical in shape. It is generally soluble in water. It contains irregular sequence of amino acids. It is more sensitive to pH and heat. For example, hemoglobin and insulin. So myoglobin, a globular protein. Functions of protein. So make about 15% of the cell, have many functions in the cell: enzymes, structural, transport, motor, storage, signaling, receptors, gene regulation, and special functions. So these are the functions of protein. So we will discuss in details every what are the different functions. Okay. So protein form a wide variety of functions in human body, essential for containing health and supporting life. So here are some key functions. Okay, the structural support. So let us see them example. So collagen provides strength and structure to skin, bones, tendons, and ligaments. Keratins found in hair, nails, and the outer layer of the skin, offering protection and support. Enzymatic activity. So digestive proteins such as amylase, lipase, protease, which breaks down food into nutrient, the body can absorb and use. Okay, and metabolic enzyme like those involved in glycolysis and the citric acid. So these are the functions of protein. Transport and storage. So this is also very important. So hemoglobin is a perfect example how oxygen is transported by hemoglobin from the lungs to rest of the body. And ferritin stores and release iron as needed, preventing deficiency and overload. So cell signaling hormones such as insulin, which regulates blood sugar level, receptors like insulin receptor, which binds hormones and other signaling molecules, transmitting signals into cell. Immune system. So antibodies identify and neutralize foreign invaders such as bacteria and viruses. Complement protein assist in the destruction of pathogens and facilitate phagocytogenesis and movement. Acting and myosin enable muscle contraction and movement. Uh, Tubulin forms microtubules that are essential for cell division and intracellular transport. Regulation and of genes expression. So we know that we will discuss in details what is transcription and all these things. So transcription factor bind to DNA and regulate the transcription of genes and histones, proteins, package and other DNA into structural unit. Catalysis and metabolic reactions. So enzymes facilitate almost all chemical reaction in the body, including digestion, energy production, and biosynthesis. Protection, as I already told, converts fibrin into blood protein, preventing excessive bleeding. This fibrinogen and skin protein create barrier that protect against pathogen and physiological damage. So this is all about the protein structure and its function. So now let us discuss about structure and function of nucleic acid. So now let us discuss about the structure and function of the nucleic acid. So first let us know what is a nucleic acid. So nucleic acids are biopolymers, macromolecule essential to all known form of life. This is the all very important unit in all animals. So they are composed of nucleotides, which are monomer made of three components. One is 5-carbon sugar and a phosphate group and a nitrogenous base. The two main classes of nucleic acids are deoxyribonucleic acid, which is found in DNA, which is DNA in short, and the ribonucleic acid. In short, it is called RNA. If the sugar is ribose, the polymer is RNA. If the sugar is ribose derivative deoxyribose, the polymer is DNA. So, what is the significance? So, nucleic acid are naturally occurring chemical compound that serves as the primary information carrying molecule in cells and make the genetic material. So nucleic acid are found in abundance in all living cells where they create, encode, and they store information of every living cells of every life form of the on the earth. So the encoded information is contained and conveyed via the nucleic acid sequence of the nucleotide, which within the molecule of RNA and DNA. So they play a especially very good role in directing the synthesis of protein. Okay. So what are the nucleobases? Basically, five nucleobases are found in nucleoacids. Okay, so nucleobases are nitrogen-containing biological compound that form the building blocks of nucleic acid, such as DNA and RNA. There are five primary nucleobases. Three are derivative of pyrimidine, and two are derivative of purine. Okay, so you can see three are derivative of pyrimidine, and two are purine. So what are the included in pyrimidine one? So uracil, that is exclusively found in RNA. Then thiamine, and that is also exclusively found in DNA. And cytosine, that is present in both DNA and RNA. So those three are the derivative of pyrimidine. And what are the derivative of purine? So adenine and guanidine. Okay, so those both nucleobases are present in both DNA and RNA. Okay, so what are nucleoside and nucleotide? Let us discuss. Okay, so a nucleoside is a molecular compound consisting of two components on nitrogenous basis. This can be purine, adenine or guanine, or pyrimidine base, cytosine, thiamine or uracil, and a sugar molecule. This could be either RNA or like ribose sugar in RNA and deoxyribose sugar in DNA. The nitrogenous base is attached to the 1' carbon of the sugar molecule. So when one or more phosphate group are attached to the 5' carbon of the sugar molecule, it is called nucleotide. So let us see what is the pictorial representation. So you can see when the sugar molecule is attached to a nitrogenous basis, we call it nucleoside. Okay, and when a phosphate group is attached here, so this is the nitrogenous basis, this is sugar molecule, and this is the phosphate group. We all three together known as nucleotide. Okay, and this two is nucleoside. Okay, so this is the best pictorial representation to distinguish between nucleoside and nucleotide. So you can see a base, nitrogenous base, and the sugar molecule is known as nucleoside. When a phosphate group is present here, we call it nucleoside monophosphate. When two there, nucleoside diphosphate, three, nucleoside triphosphate. So those are the examples of nucleotides. So this is the overall pictorial representation. Okay. So nitrogenous bases and the pentose sugar from nucleoside plus a phosphate group from nucleotides, and then all the nucleotides combine and gives the nucleic acids. So, what is a polynucleotide? A polynucleotide molecule is a biopolymer composed of 13 or more nucleotide monomers covalently bonded in a chain. So, DNA and RNA are example of polynucleotide with distinct biological function. The nucleotides can be held together by phosphodiester linkage to form polymer. So, this phosphodiester linkage is the most important bond for all human body and mammals. Okay. So what is phosphodiester bond? A phosphodiester bond occurs when exactly two of the hydroxyl group of phosphoric acid react with the hydroxyl group on another molecule to form the two ester bond. So you can see, so this OH group present in the sugar, which is making a bond with the phosphoric group present in the 5' position of another sugar, and this is making a bond, diphosphodiester bond. Okay. The phosphodiester bond are the central to all life, as I said, on the earth, as they make up the backbone of the strand of nucleic acid. If this bond is not forming, the nucleic acid will not form and there will be no life. So in DNA and RNA, the phosphodiester bond is the linkage between the 3' carbon of one sugar molecule and the 5' carbon of another. So deoxyribose in DNA and ribose is RNA. So now, what is the overall structure of DNA? Deoxyribonucleic acid. So you can see it's two strands coil each other, and by different hydrogen bonding interaction between the bases present in the two different polypeptide strand, it gives helical structure. So you can see this is one polypeptide chain, this is another polypeptide chain. So you can see a hydrogen bond is forming between the different nucleobases present there, and it is giving a helical structure. Composition of DNA. So DNA is molecule composed of two polynucleotide chain that coil around each other to form a double helix carrying genetic instruction for the development, functioning, growth, and reproduction of all organisms and many viruses. So what is So in all species, it is composed of two helical chain bound to each other by hydrogen bonding. Both chain are coiled around and in the same axis and have the same pitch of 34 angstrom or 3.4 nanometers. The pair of chains has a radius of 10 angstrom, okay, or 1 nanometer. According to another study, when measured in the different solution, the DNA chain measure 22 to 26 angstrom. Okay, so yeah, this is the overall composition of the DNA. So variation of DNA. So most of the DNA in the classic Western Crick model, which is the common structure, simply called bDNA or B form of the DNA. In certain condition, different form of DNAs are found to be appeared like aDNA, zDNA, cDNA, ddNA, eDNA. This deviation in forms are based on their structural diversity. So, what is the reason or what are the factors responsible for structural variation of DNA? So, DNA, the genetic information carrier molecule of the cell, is a long polymer of nucleotides and can adopt different types of structural conformation. The various type of conformation that the DNA can adopt depend on different factors such as hydration level, salt concentration, DNA sequence, quantity and direction of supercoiling, presence of chemically modified bases, different type of metal ion and its concentration, presence of polyamines in solution. Different form of DNA. So X-ray analysis of DNA crystals at atomic resolution have revealed that DNA exhibit much more structural diversity than formerly envisaged. Okay, such variations are bDNA, aDNA, zDNA, cDNA, ddNA, eDNA. So those are the some examples where this kind of structures are found. Okay, this is not that much very important to remember. Okay, just good to know. So these are the some pictorial representation. So you can see this is the ADNA. Okay, this is BDNA, which is the classic one, and this is cDNA, and this is zDNA. And those cavity are known as groups. Okay, so this is minor groups. So major groups. Okay, I will explain. So composition of RNA now. So RNA is a polymeric molecule essential in various biological roles in coding, decoding, regulation, and expression of genes. Each nucleotide in RNA contains a ribose sugar with carbon number 1' through 5'. A base is attached to the 1' position of the sugar. In general, adenine, cytosine, guanidine, uracil, and guanidine are purine derivative and uracil are pyrimidine derivative, as I already told in the previous. So this is the overall structure of RNA. So you can see this is the sugar molecule. This is the nitrogenous base attached to the 1' position of the sugar, and this is the phosphate group which is attached to the 5' position of the sugar. Okay, so this is the structure of RNA. Right? So three main type of RNA or protein biosynthesis. There are three type of RNA found in protein biosynthesis. One is mRNA or messenger RNA. This type of RNA functions by transferring the genetic material into the ribosomes and pass the instruction about the type of proteins required by the body cell. So based on the functions, this type of RNA is called messenger RNA. Therefore, the mRNA plays a vital role in the process of transcription or during the protein synthesis process. tRNA means transfer RNA. The transfer RNA is held responsible for choosing the correct protein or the amino acids required by the body in terms helping the ribosome. It is located at the endpoint of each amino acid. This is also called as soluble RNA, and it is formed link between the messenger RNA and the amino acid. So rRNA or ribosomal RNA. So this rRNA is the component of ribosome and are located within in the cytoplasm of the cell where ribosomes are found. In all living cells, the ribosomal RNA plays a fundamental role in the synthesis and translation of mRNA into protein. The rRNA is mainly composed of cellular RNA and are most predominant RNA within the cell of all living beings. So this is the structure of messenger RNA. This is the ribosomal RNA. This is the transfer RNA. What happened? This transfer RNA bring different amino acid based on the coding in the messenger RNA. I'll discuss all this thing coding, decoding, and this hold this template and allow to sit this amino acid here and make the peptide chain. So what is the main difference between GNA and RNA? This is very good to know. So main difference is sugar unit. Okay, so here deoxyribose sugar in DNA and here ribose sugar in DNA. Okay, generally DNA is double stranded. Okay, and two strand are attached to each other by hydrogen bonding, and generally RNA is single stranded. Okay, so thiamine exclusively found in DNA and uracil exclusively found in RNA, and the common bases are cytosine, guanine, and adenine. Okay, so these are the key differences between DNA and RNA. We can see this is the more clear presentation of difference of DNA and RNA. Now discuss about what is genetic code and codon. This is very important to know. Okay, so the genetic code is a set of rules by which the sequence of nucleic acid in DNA and RNA is translated into the sequence of amino acid in protein. It is universal language that dictates how the four-letter alphabet of DNA and RNA, okay, is translated into the 20 letters alphabet of protein. So key features of genetic code. So triplet code. The triplet code is read in sets of three nucleotides, okay, known as codons. Each codon specifies one amino acid. So redundancy, there are 64 possible codon, but only 20 amino acid, meaning that multiple codon can code for the same amino acid. This is known as redundancy and degeneracy of the genetic code. Start and stop codons. There are specific codon that signals the start. AUG, that is the start codon. Actually, I'll show the graph. Okay, and stop codon EUA, UAG, and UGA. Okay, these protein are in the potency. So universality, the genetic code is nearly universal, meaning it is same for almost all organisms, from bacteria to human. Types of codon. There are three different types of codon. One is sense codon, codon that sense specific amino acid. So there are 61 sense codon. Start codon, which is starting the synthesis of amino acid. So this is the AUG is the start codon, okay, which initiate translation and codes for amino acid like methionine. Okay, stop codon, codon that signals the end of the protein synthesis. This UAA, UAG, and UGA, those are the stop codon. The genetic code and codons are crucial for the process of translation, where the mRNA sequence is read by the ribosome and translated into a polypeptide chain, which then folds into functional protein. Understanding the genetic code in fundamental to genetics, molecular biology, and bioinformatics, and has application in genetic engineering, biotechnology, and medicines. So this is the overall graph of the codons. Okay, so you can see total 64 genetic codes and 20 amino acids you know. So we can understand that one amino acid may have several code. Okay, so you can see we can just arrange like nucleobases like this: uracil, cytosine, adenine, guanine, like this in the this axis, and here also UCAG, and here also. Okay, then if we take this one UUE, so U and U, so we can take this kind of codon. Again UUC, we can get this kind of codon. So these are the different codon we can make based on this table. Okay, and we can see this codon is corresponding to phenylalanine. So phenylalanine will sit in the messenger RNA which have this kind of codon. Okay, UAUA, UAC, UA. Similarly, leucine will have corresponding to those codon. Okay, actually leucine has the highest number of codon, and they can sit in many places. Okay, okay. So we can see these are the different amino acid with different codon. So here we can see uh, where is it? Thionine? Okay, thionine. So methionine, sorry, this is the start codon. This AUG, this is the start codon, and methionine amino acid only will start the synthesis of protein. Okay, and stop codon, you can see there are few stop codon. One is UGA. Okay, those are the stop codon. This will stop the UAA, UAG. Those are the stop codon. It will stop the synthesis of amino acid. Now let us discuss how this dictate the synthesis of protein. Okay, so if we have a DNA structure, okay, so we can see this is, if we consider this part, so this has this kind of different nucleobases. Okay, thymine, adenine, cytosine, like that. Okay, so the transcription mean it will arrange the different amino acid in different codes and triplet codes like AUG. Definitely AUG will start. Okay, then these are the different codes. Then the translation. Okay, this is messenger RNA template actually. Now the amino acid will come and seek to the specific codon assigned for them, and they will give different amino acid sequence, and then protein synthesis will take place. So now let us talk about what is transcription. So transcription is the process by which the information in a strand of DNA is copied into a new molecule of messenger, okay, RNA. So transcription is the first part of the central dogma of molecular biology, DNA to RNA, how it is making, and it is transfer of genetic instruction in DNA to mRNA. Okay, during transcription, a strand of mRNA is made to complement a strand of DNA. You can see how it happens in the diagram. So you can see this is the one strand of DNA with different nucleobases like adenine, cytosine, thymine, like that. In transcription, in messenger RNA, so this is the messenger RNA. We can see they are making a template with different codons. So those are called codon, three bases. Okay. So now this will correspond to a specific amino acid which can sit here and make the polypeptide chain. Okay. So what is translation? So in molecular biology and genetics, translation is the process in which ribosome in the cytoplasm or endoplasm reticulum synthesis protein after the process of transcription. DNA to RNA in the cell nucleus. The entire process is called gene expression. Translation proceeds in three phases: initiation, elongation, and termination. The translation proceeds in three phases: initiation, the ribosome assembles around the target mRNA. The first tRNA is attached at the start codon AUG, the methionine amino, and then elongation. The last tRNA validated by the small ribosome subunit. Okay, transfer the amino acid it carries to the large ribosome subunit, which bind it to the one of the proceeding admitted to transcript RNA. The ribosome then moves to the next mRNA codon to continue the process. This is called translocation. Okay, and the creating then amino acid chain, and termination. Finally, when it stop codon is reached, the ribosome releases the polypeptide chain, and it stops the synthesis of protein. So this is the overall pictorial representation we can see. So ribosomes are complex molecular machines found within the living cell. Okay, they are served as the site of biological protein synthesis, overall synthesis. Okay, the ribosome read the sequence of messenger RNA. Okay, this is the messenger RNA. Okay, and translate the encoded information into the specific sequence of amino acid to form polypeptide chain. This process is vital, okay, for cellular function and life. So you can see ribosome consists of two units. One is small subunit, another is large subunit. Okay, so this messenger RNA is sandwiched between the large and the small subunit. Okay, now what the tRNA is doing? tRNA is bringing different amino acid and sitting the assigned codon. Okay, then when the stop stop codon is coming, it will get released, and the protein will come out from the ribosome cells. So we can see the function of ribosome. What is the function of ribosome? The primary function of ribosome is to synthesize protein through a process called translation. This involves several steps. Initiation, the small subunit bind to the mRNA and identifies the start codon. The initiator or tRNA binds to this start codon. Okay. Then now second is elongation. The large subunit join to form a complete ribosome. Transfer RNA molecules bring amino acid to the ribosome in accordance with the codon sequence on the mRNA. The ribosome catalyzes the formation of peptide bond between the amino acids. And now final step is termination. When a stop codon on the mRNA is reached, the newly formed polypeptide is released and the ribosome subunit dissociates. So what is the major and minor groups in DNA? So DNA double helix has two distinct groups that play crucial role in the molecular biology. So one is major groups, another is minor groups. Major groups and minor groups. Okay. So these groups are formed by the helical structure of the DNA and significant because they allow protein to interact with the DNA molecule in sequence specific manner. So structure and formation. The major and major groups are results of the angle at which the bases attach to the sugar phosphate backbone, creating asymmetry in the double helix. So what is major groups I told? So major groups, this groups is wide, about 22 Armstrong, and differ. It provides a large surface area where protein and other molecules can interact with the DNA. And minor groups, okay, these groups is narrower, okay, and shallower. Although it is less accessible than the major groups, it is still allow the specific protein DNA interaction. So why these groups are important? So when we are taking like some cells, DNA cells is infected with cancer, okay, and we need to break the DNA of those cells. At that time, we will take some chemotherapeutic drug molecule, okay. So those chemotherapeutic drug molecule, what they will do, they will bind in the major groups of the DNA, okay, and then they will disrupt the overall structure of the DNA, and they will break the DNA of that particular cell. So the cancer cell will not be able to replicate, or it will not grow, and the cell reproduction will stop, and it will stop the cancer. there are several chemotherapeutic drugs are available, which can act as the DNA binding agents. Okay, there are several chemotherapeutic drugs like transplatin. Okay, then Taxol, and then many more. Okay, so they actually bind to the DNA to the cancer cell, which is present in the infected cells. Okay, so if they bind, they will stop the formation of more cells, or the formation of the growing of the cells. Okay, so this is all about the structure and functions of nucleic acids. Okay, so thank you for listening. See you in the next class.